Hi, my name is Dr. Jesse Kleingardner, and I'm an assistant professor here in the Department of Chemistry and Biochemistry at Messiah University. I teach general chemistry and biochemistry, and I also mentor a number of research students in my research laboratory here. They have a variety of majors, but they all have in common that they want to learn science by doing science. Proteins are versatile biomolecules that carry out most functions in the cell. That includes signaling, transport, catalyzing chemical reactions, turning some biomolecules into other biomolecules. It also includes regulation, copying DNA, transcribing it to RNA, you name it. Most functions are accomplished by proteins. So they're versatile biomolecules, and our ability to design customized proteins with customized functions has led to advances in medicine and biotechnology, and we are making rapid advances in that field. I aim to sort of add my own research to that field, and in general, the research that I research with students falls into the category of metalloprotein engineering. So engineering proteins, customizing them, and designing them, but particularly ones that have metal ions, mainly. So the idea is to modify, to customize, to design proteins that have desirable properties or functions, structures, etc. The types of proteins that I work with, most of them are heme proteins, and the idea is to turn non-heme proteins into heme proteins that have unique structures, or to substitute the central iron in the heme group for other metals, or to incorporate unnatural amino acids, or switch out the metal binding amino acids. And the idea is to be able to rationally change the properties of the proteins so that they can catalyze reactions that are important to renewable fuel production or fuel usage in, for example, fuel cells, that sort of energy conversion. Another type of protein that we work with engineering in the laboratory are fluorescent proteins. And we do a couple of things. One thing we try to do with fluorescent proteins is to design metal binding sites to be able to bind specific metals in the fluorescent protein so that we can use it to fluorescently detect metal ions inside the cell in real time, dynamically. We would be able to see then the changes in metal ion concentrations over time in response to stimuli, etc. We're also working on trying to tune the properties of the fluorescent protein in order to be able to detect them easier, to make them more useful for biochemical applications. Finally, in collaboration with the engineering collaboratory here at Messiah, we work on designing an HIV binding protein that's been fused to a fluorescent protein. And the idea here is to be able to fluorescently detect HIV in blood using like an inexpensive point of care device that can be utilized in third world countries. All of these projects involve a number of core biochemistry techniques that involves gene manipulation, working with DNA, changing the genetic sequence of a region of DNA. We also work on taking that DNA that encodes for a protein and actually producing that protein. We use E. coli cells for this, called recombinant protein expression. And we purify the protein using biochemical techniques. We also are able to characterize proteins. We can characterize their melting temperatures. We can characterize them spectroscopically, and we can determine their size or binding via gel electrophoresis, and even are getting into some electrochemistry to be able to measure catalysis. I am certainly happy all the time to talk about research, to answer questions about what I do and how I incorporate scientific research into the education of students here at Messiah. You can find my email, jkleingardner@messiah.edu, on the Department of Chemistry and Biochemistry website at messiah.edu. And you can also find my website and check out some more information on the research that I do here. Thank you.